3, 2, 1. Hi, welcome everybody for this class of genomics. And today I'd like to start the session with hopefully a quick COVID-19 update. So as you know, I'm pulling the data from the sewage. So here this is the amount of signal that they can detect in the sewage of the Greater Toronto Area. We were around here at the beginning of the back in person types of class, and so it looked like this is going to flatten, but in reality we're at the beginning of a new wave. So the main protagonist of this wave is the variant BA2, as you probably know. So that's a variant of Omicron that is even more infectious than Omicron, that is about the same level of severity as Omicron. It's just another variant of Omicron. So bad news is that it's substantially more infectious. People say 80%, and Omicron was already extremely high. So this one is even more contagious. The good news is that it's not more deadly than Omicron. It's the same. And so, of course, you can notice that there is a very good correspondence between the beginning of this wave and the removal of the mask mandate. And that has been seen in many countries in Europe. If you've been following the numbers, for example, in the Netherlands, France, the UK, or Austria, as soon as people remove the mask, then they got that wave. So we can know a little bit what's going to happen if we look at, for example, the profile of France, which has followed. Canada has followed more or less the profile of France. And so the wave in the case of France went about halfway through what was in December. So that's what we can prepare for. Like this wave is probably going to be bad, probably going to be less bad than the one that was in December. But we had planned, we were discussing with Theodore whether we should do some sort of socializing activity for BIOD25, and I would love that. But I don't think it'd be very responsible to plan for something in 1 or 2 weeks. So very unfortunately, in class will be the only kind of social activities that we're going to have. And I would definitely love to go for a beer, go for pizza, or whatever that is with you guys. But I don't think it'd be very responsible given the situation of the epidemic at the moment. So that's it. The wave has started. We're not going to change anything in terms of the class. It's just to remind you to be responsible. You're good citizens, to continue good citizens. The point I want to make here is that already there is a beginning of a visible signal on the hospitals. So the yellow curve that you see here is the number of people who go to the hospitals but not to the ICU, and this has started to rise again. And this is Ontario, the whole of Ontario. It's not really visible here, but it seems that the trend in ICU has stopped decreasing, so it's going to probably stabilize here and then rise a little bit. And of course, ICU are the people who are with very severe COVID symptoms. So that's already relatively bad, that the hospitals are starting to fill up again, but that's the way it is. And so one thing that I'd like to remind you is that we have to approach, at least in this class, at least in BIOD25, every problem like a scientist. Okay, we're not panicking, we're not taking this with our emotions, saying this is a scandal, this is good, this is bad. So we're trying to think about the bigger picture like scientists and in terms of quantitative terms. And one thing that I'd like to remind you, and I've said that multiple times and I will keep saying that, is that the real problem of COVID-19 is the effect on the hospitals. And so for every person who goes to the hospital and survives COVID-19, that's great, but that's people who could not go to the hospital and who had life-saving surgery that was scheduled or something like that. So you have to look at the bigger picture of what's the effect of COVID-19 on the society. And so one way you can do this is with a graph which is called excess mortality. So that's what you can see here. These are the dates, these are the weeks from 1 to 53, and the years are indicated by colors, as you can see here. So back to 2014, oh, what a great year, 2015, 16 as well, those were the days. So you can see that the level of mortality was approximately here in the whole of Canada, so that's the number of deaths per week. So for you to know, people die more in winter in Canada, so that's this month here, and there than in summer, and that is because of all the diseases that are associated with winter. So that's typically a curve that looks like that. And the dreadful year is 2020, the one in pink here, and you can see that there is this peak around March, and that's the beginning of COVID-19 in Canada. So if you look at the number of people who were officially dead of COVID-19 in Canada, that's approximately 8,000 within this 2 to 3 month period around March. And they're all here in the excess mortality in the pink curve. Okay, like if you make the difference between 2019 and 2020, there are approximately 8,000 excess dead people. So far everything is accounted for. The issue though is that when you look at the curves, is that over the year, like 2020 is here in pink, now we're above the threshold of 2019 and 2021. The data is still not completely final, so this is data from the government of Canada that I'm pulling, is above the level of 2020 here. 21, you can see that's above the level of 2020. So even though we are under the impression that things are going better, because they are going better in terms of the epidemic, it's not necessarily the case in terms of hospitals. And one thing that you can look at, so this point here is probably aberrant, but if you look at 21, last year, in the period of August, for example, there are approximately 2,000 excess dead people. But that's not a month where there was a lot of COVID death. There are only 300 deaths in COVID in 2021, with COVID in 2021. The excess mortality, where is it coming from? Different sources, but one of them is just the fact that the hospitals have been severely impeded by the epidemic. And these are people who cannot get the treatment that they deserve, that they had to have, and that they would have had without the epidemic. So one thing that I just want to remind you is that the way to approach this epidemic is not really thinking about yourself, thinking about the effects that they have on people that you may infect or who might infect you. It's really about the hospitals. There are more people dying of the fact that the hospitals are not able to take them than people dying of COVID-19 at the moment. And that's why be aware of that. Be aware that the issue is really try to not go to the hospital, try to not send anyone to the hospital, and spread the word, right? That this is really about saving our hospitals and making sure that they can take care of the people who need it. Enough for that. Okay, so other topics, a little more joyful, but not that much. Course evaluation, I think at some point, maybe you have already, will receive a request to evaluate the course. So the way it works, I found it on Reddit, people were quite, I don't know, they had questions about why do we evaluate the course, why teachers say, you have to evaluate my course, please, please, please do it, and they're practically begging that you guys would evaluate it. So the way it works is that there are a standard set of questions, and the registrar is asking me to choose 3 extra questions that I'm adding on top of the usual questions. So the usual questions are for the registrar to know how I'm doing, and that's the way they evaluate me for my tenure or whatever they want to evaluate me for, just to make sure that I'm being a good teacher. And the other 3 questions, the one that I'm choosing from a list of really a lot, there are at least 500 items. This is for me, just to know how to improve my course, etc. So I'm interested in every kind of feedback that you have. If you want to be super mean or evil, it's fine. Don't worry. I think I'll survive. I can take it. If you want to be friendly and nice, it's also fine. I like it. But mostly, I want to know how to improve the course. This is not the last year I'm going to give it, so think about students next year, and then think about practical things that you could tell me to improve the course. If you don't know how to improve it, but you know what you didn't like, well, go ahead, tell me. That's always good to hear. Okay. So the reason why most teachers are really insisting a lot about asking the students to evaluate them, I suppose, is because it's required for the tenure, and they want to make sure that score enough points because of the department. They're probably looking at that very closely. But my department, as far as I'm aware, is not looking at that very, very closely. This is not the main way that they evaluate the quality of my teaching. Okay, so if you feel like it, just give me some feedback. I promise I'll read it all with attention. If possible, I'll try to improve if the suggestions are practical enough. So another thing that I thought was probably important to talk a little bit about is this moment 2 weeks ago when I spoke about Adam and Eve, and I realized that it's been somewhat confusing for some of you, especially if you're religious, and if Adam and Eve are characters in your religion, then there was a bit of a questioning, maybe not a mismatch, but at least some question whether it would infirm, confirm, or be completely independent of that view of Adam and Eve in the Bible, for instance. So I'd like to come back to this example and then give another one that will probably help you understand a little bit better what that means by the most recent common ancestor of the Y chromosome and of the mitochondrial DNA. So the most recent common ancestor has nothing special. So let's look at this family tree here. This Silvio, the grandfather, who has 2 sons, Fabio and Vincent. So he's passing his Y chromosome to Fabio and Vincent. And then Fabio has 1 son, Dario, and Vincent has 2 sons, John and Robert. So obviously these guys remain in Italy, these guys move to the US. And so think about it. So once Silvio is dead, Fabio and Vincent are dead, only the grandchildren are left, Dario, John, and Robert. So who's the most recent common ancestor? That's Silvio. Okay, Silvio is like the most recent common ancestor of them all. That's the Adam of the Y chromosomes in that mini scenario. Okay, but if Dario dies, who is the most recent common ancestor? Okay, like the only people who are left are John and Robert, and the most recent common ancestor is Vincent. So just the fact that Dario died switched Adam from being Silvio to Vincent. So there's nothing special about Silvio, neither about Vincent. It's just that depending on who's left at the moment when we watch the family tree, there's just one person that has to be the most recent common ancestor. There has to be one. Okay, this is a mathematical absolute obligation, and this person has nothing special. It just happens to be the guy who is the one who gives kind of a descent that's present into this population. So if Dario dies first, then the most recent common ancestor becomes Vincent, which really shows that Silvio had nothing special. It was not really about Silvio. Yes?
Okay, so just to make sure I'm understanding clearly, so the most recent common ancestor would depend on who's alive. Is that it?
Yeah, the most recent common ancestor depends on who's alive, he says. Yes, that's exactly it. Okay, so depending on who's alive, that changes who's the most recent common ancestor, because in reality, what you're asking is the most recent common ancestor of whom exactly? And when we say that, we say of everybody who's alive on the planet when we talk about Eve or Adam. All right. So the MRCA, the most recent common ancestor, has nothing special. In the case on the left, when only the grandchildren are alive, the most recent common ancestor is Silvio. But if Dario dies first, I meant Fabio. I meant Dario here. If this branch disappears, Fabio and Dario are dead. Then it becomes Vincent. Okay. For the most recent common ancestor of all Y chromosomes, that's Adam, or mitochondrial DNA, that's Eve. The same applies. The Adam and Eve of 2022, of today, will not remain the same, missing keyword, forever. With time, they will change. There's nothing special about the current Adam and the current Eve. When part of the population disappears, let's say in 2,000, 3,000, 100,000 years, the most recent common ancestor of everybody who's alive will not remain the same person. They will move forward, just like in this case. So it cannot have anything to do with anyone special. There's nothing special about that person. It just happens to be the lucky guy that we pick in that example. Okay, so that cannot be anything related with the Bible or with any other religion, because these are moving Adams and moving Eves. And so that was not very clever, of course, from these guys who made this study 10 or 15 years ago to call it Adam and Eve, because it's extremely confusing. It has nothing to do with anyone special. They did this just because of marketing purposes, or maybe because they were religious. And in retrospect, that was maybe not so smart from me to call them Adam and Eve. Maybe I should have not even talked about this too much because it's confusing. But I hope you get the point that this is something that's just the fact that if there is a family tree, there has to be one guy who's on top or one lady who's on top. And that's just because of who's alive now that we define that's the most recent common ancestor. When the people alive change, that also changes the most recent common ancestor. Right? Is that clear? Is there a question? Good. Okay. Yes.
So like looking at it, it doesn't matter who my great-great-grandfather was. If like one of his grandchildren happened to be dead, then he's not the most common recent ancestor anymore.
Yeah. So the question is, doesn't matter who the great-great-grandfather or great-ancestor was. If all the children are dead, then he cannot be the Adam. Yeah, exactly. That's the point. It's just, do you still have descendants alive? Yes or no? Yes, you can be the most recent common ancestor. No, you're out of the game already. All right. I know that these concepts are new for you, that you're not manipulating trees a lot before this class. So I know that this is a little bit hard, but it's not crazy complicated once you think about it. Okay, blog post. Thank you for your title and TLDR. There was some really good things. I wanted to share with you some of the examples I like. I hope that the authors will forgive me for sharing that, but this is because I like them and I keep them anonymous anyway. So these are two examples that I thought were kind of nice in the sense that they completed the assignment in the way that I expected it. And I've picked one that is in the short range, one that is in the long range, to show that you really have both options. You can go short, you can go long, and it's not really making it better or worse, the length. It's really about the content. So I like this one, forensic technique, personal bacteria as the new fingerprint, question mark. Bacteria can be found everywhere, but did you know we each have different bacterial colonies living on our skin? With the help of their distinctive qualities, perhaps they can help us solve crimes. So this is a good example because it has everything it should have. There is the keyword, forensic here is very important. Bacteria, of course, and kind of fingerprint as well contributes to being keywords. So everything is in the title. You also have a question in the title that's already appealing. What's the relationship between bacteria and a fingerprint? And then in the TLDR, this is really compact, like it's only a 2-sentence TLDR. And that kind of, the key is revealing, but not too much. That tells us that, like really, we all have different bacteria on the surface of the skin. That's really telling you something about the story, about the paper, what the authors are going to tell. But you don't tell more, okay? Because otherwise that'd be kind of a summary, etc. So you want people to read the article. And so that's enough of a reveal here and enough of telling what the article is about, but you're telling what's inside the article about. So people who just spoke of, who told what the topic is, that's not sufficient. I think when that was the case, I've written a comment saying that's not sufficient. So the TLDR is not just to say this article is about bacteria. That's not sufficient. Like you have to tell a little bit what's going to be inside and give a bit of a primer. Okay, so another one which is kind of more on the long side, and I've chosen these two because they were quite at the end of the spectrum. The butterfly effect, what leads to the wide discrepancy in transmission between SARS-CoV and SARS-CoV-2. Although SARS-CoV and SARS-CoV-2 indeed have very similar genomic components, why do they exhibit such remarkable differences in terms of their spreading capacity and toxicity? A recent structural proteomics study points out that the underlying causes could be subtler than you think. The potential butterfly effect that starts with a few minor changes in the molecular architectures of the viruses. So that's kind of really at the limit of the length that you should have for TLDR. Longer than this would be too long, but that's just right. Okay. Well, the title is also really pushing it a little bit in terms of the length. Like you should try to make it a tiny bit shorter, but that's fine. I mean, in terms of readability, that's a bit less good, but in terms of machine indexing, that's kind of nicer to have long titles because you have more keywords, like butterfly effect, SARS-CoV, SARS-CoV-2, etc. Okay, and again, it's about the reveal, that tells something about the article. The butterfly effect is not just telling you, I'm going to tell you some generic information about SARS-CoV and SARS-CoV-2. You really have something more concrete, and so that's why these were two examples that I thought would be useful for everybody to read. In general, to get back to the grading and to the way I did this, so I really calibrated it. Most of you got the titles right. Like there was no particular problem with the title. It was more with the TLDR. And so the rule is that if you got 2 or more out of 3, then that's okay. If you got less than 2, that's not okay. You need to make serious changes for your final blog post. And so I've taken the decision to hurt you a little in that case, to give you 1.5 or less out of 3, and hoping to make you a little bit angry to get your attention. Okay, in the sense that I want to get your attention now to say this is not the way you're supposed to write the blog post, and I'm taking like 0.5 points away from you now because it's very short, like it's very small stake. Remember that the blog post is actually 30 points. And so I don't want you to make the same mistake out of something that's worth 30 points. So I want to get your attention now if I give you less than 2. And you should not really look too closely about into the grading, like, okay, he took a point for concreteness, etc. It's like if you got less than 2, it means probably you're a little too vague. Okay? And I don't want you to write the article this way, because if you do it this way for the whole article, then it's going to severely lower your grade. So I'm giving you a signal, which I know is a little bit painful, and I hope one day you'll forgive me for doing that. And I hope that when you get a 30 out of 30 in your final blog post, you'll forgive me. But I need to get your attention that very often the TLDR was too vague. This is not the way it's supposed to be. It has to be concrete. The whole blog post has to be concrete. Okay, if you have an issue with that, I remind you that I have office hours, you can challenge my grading, of course, and I'd be happy to see this with you, but only during office hours. And again, I remind you that it was only to get your attention when I thought this is a little too vague, and I do not recommend that you write the whole blog post this way. Any questions on that? Okay, let's move on then. So right now you should be starting to write already. That's my advice. Of course you have time. There is time even after the last class, etc. But you'll see writing is long, difficult, and it always takes longer than you think. So the earlier you can start writing, the earlier you can start thinking about the way you're going to structure your blog post, the better. The example that you'll find in Sarah's little explanation, you'll find it in many places. That's called the inverted pyramid, and that's the way you have to write the blog post. That's also the way you have to write pretty much everything in your life from now on. There's not really any exception. So the inverted pyramid is the template you have to follow for this kind of blog post. Bear in mind that your readers have little time and that they will drop after 1 or 2 paragraphs. So you want to put the most important first. That's the idea, is that remember it's a free world. You compete against Instagram, YouTube, Facebook, the next class, or sleeping. So most people will give you their attention for just a little time, but at some point they'll get bored. And so if you have put like the most important thing at the very, very end, chances are that they will not even get there. So you do it in the opposite way. You really put the crucial things first. And that's why we have the TLDR. Is that even if they stop at the TLDR, they have learned something, which is not just the topic of the article. The most important does not have to be the main finding of the article, and that's very important, okay? The conclusion of the author does not have to be your conclusion. It does not have to be exactly what is the main important in your case. Okay? Especially if it is a little vague. Sometimes the authors themselves or methods or something like this, they don't have something very concrete to say. They say, yeah, we've got a method that can do that. So it can be the question, actually that's the most important. Sometimes even the fact that there's a question is something that we didn't know. You know, we thought it was a solved question, and then these guys say, no, that's a completely open question, nobody knows how it works. That's really very interesting. Okay? It can be the experimental system as well, sometimes, especially in the case of a method, that's actually the most important. It's not really what they got with it, but the way it works, or maybe the key that they introduced compared to the state of the art, or the fact that there is a debate. Okay, sometimes people come up with one idea, but this is actually not the only one to explain something, and it can be very informative for the reader to know whatever I'm going to say now, there's no agreement. Okay, so take a little bit of distance compared to the text, to the article, and then remember that the most important in your blog post does not have to be the very final sentence of the paper. Very often this final sentence is a bit of a trap on top of it, because people say, yeah, and with that we're going to cure cancer, which is never true. So be careful if you have a paper like that that says, thanks to our beautiful advances, we will be able to cure all the diseases, because I will be okay with that for the grading, but I will not allow this to be published on the blog. We already had some issues of people who complained because some of the readers actually have the disease you're talking about. Okay, and so in that case, it's really important to remember that we don't want to give false hopes. These are people who are actually looking for treatment, who cannot understand medical doctors or more complicated sites, so they do end up reading blog posts because this is usually the level that they can understand. And if the blog post is saying you can be saved if you just use that treatment with this new single cell, whatever that is, it's actually pretty bad. So you have to be fighting disinformation and actively protect yourself and protect everyone from doing this kind of stuff. So don't jump to conclusion. Don't assume that the very last sentence of the article is the best and that should be your conclusion. Right? Is that clear? Okay. Now, one thing is that I'd like you to contact the authors, and the best moment to do this is 2 or 3 weeks ago already. So do this fast, because that's important that you get time to contact them to see whether you can get that. So if you can, that's great. If you cannot, that's fine. Don't insist. But that's something that I'd like you all to do. And if you can get that, you're going to get bonus points on the blog post. You can still get a nice grade without this. Don't worry about this. So how do authorships work in biology? Every science has got a different code of conduct and different, let's say, culture about authorship. And so you have to understand a little bit how that works. So that's one article that I'm taking as an example. That's the one that I treated myself and that I gave, that is the demo post. You can see that there is Isabel, she's the first author, and there's also Fernando here. And both of them have this little envelope that you can see next to their name. It means that they're corresponding authors. It means that they're responsible for the content of the article after its publication. It also means that there are people who are who agree to be contacted to talk about the article. So both of them are contact people. Yes?
For my article, there's no author with a corresponding thing.
Yeah.
And then when I look them up, there's no email either.
Yeah. So, question is, for the article, there's no envelope, there's no symbol, and when you look up, there's no email address. Yeah, that's possible. I'll get to that in a second. So the culture in biology is that this person here, the first, is the one who did 99.9% of the work. This person here is the boss, person who does nothing at all but pays. So come on, like, if you pay, you get to be last and to get the little envelope next to you. And people in the middle, they did nothing. They're just friends, cousins, etc., whatever that is. Or people who have enough, you know, like you owe me a beer, you don't need to pay the beer, just put my name on the article because I need to defend my PhD and we're good. So it's not really clear what people in the middle do in general, but I'm exaggerating, of course you got that. There's this idea that only the first author did the work, basically, and everyone in the middle are guests at the party. Right? And this person didn't do anything except getting the money for it, which is highly regarded in biology. Like, you consider to be the boss if you're the person who gets the money, because you have to read the project, think about it ahead, and then also secure the resources and get the infrastructure. So in biology, which is an experimental science mostly, these people are highly regarded, and they also put themselves last out of courtesy, in a sense, for the first author to be more visible. Okay, so that's the norm, but it's not always the case. And in general, you will see when there is no symbol, the person who wants to be contacted is the last, if it's a biological paper, which I think is the case for all of you. Okay, so without this symbol here, it would be Fernando anyway, the contact person. That's the boss. If there's no email address, it means that the people are not super eager to be contacted. They didn't want to leave their email address, and so why is that? It can be that they really don't want to be contacted by anyone because they just don't like. Or it could be that they're fed up of their emails being scraped by spam bots on PubMed. You know, you not believe the number of spam email I receive for all sorts of things because people know that I'm the author of that paper. Then they craft spam, but also more elaborate scams, inviting me to conferences that don't exist, etc. Just because they know that I'm going to respond. This is my work. So some people get a little tired of that, and they don't want their email to be necessarily very visible. Okay, so if they don't put their email, it doesn't mean that they necessarily don't want to be contacted. Sometimes it's like they want to reduce a little bit the spam in the world. But anyway, you can find their email because they will give an institution, and so you can Google their name and the name of that institution and see what's their lab. There's a phone number. Usually there's a contact. Like, you will find a way to contact the last person. I've never found a case where it was impossible with Googling and just looking at the registries of the institutions where they work to find the addresses of these people. Scientists want to be accessible, almost all of them. They leave a way to be contacted about their work, also because they hope that you'll be a very famous journalist, which you're not, unfortunately for them. Okay, so you will find the name for the address for that person almost all the time, but sometimes you need to investigate. But like I said last time, try to hit for the first person, not the last. The first person is going to be more friendly. So try to hit that person first. So the corresponding author is the person who leaves her contact details for all inquiries about the paper. There can be more than one, usually indicated by a star or an envelope. The last author is usually a professor or principal investigator. The first author is usually a student or postdoc, doing everything. My recommendation is that you write to the first authors, even if they're not corresponding authors. Okay, in that case, she left an envelope, but even if they don't, try to find their contact anyway. And it's going to be on the website. Finding their contact is usually easy. Write from your academic UTSC address, don't write from your Facebook or something like that. Introduce yourself briefly, be very polite so that you really, really, really enjoyed their work. That's very important. Scientists are extremely gullible. As soon as you say, oh, I love your work, then you can get anything out of them. You should try this on me, you get max grades all the time. Be very polite, okay, you enjoy their work, and be specific about why in particular. Okay, like you don't say, I just love your work, I say, I love figure 3 because it was the best. Say you write a blog post and you like the authors to give 1 or 2 sentences about the most difficult challenge they had to overcome. That's my recommendation, just to break the ice. Instead of saying, tell us something about the article, it's too vague. Just say, okay, what was really the hard part for you? And they will love to speak about this because they're really proud of what they managed to overcome. So, and these are things that are not in the paper in general, so that's going to be added bonus because they're going to give you the story of how they sorted out the most difficult problem, and they're going to be happy to explain that. So good luck with that. It's not an easy task, but it's a fun one. All right, so let's go right into genome editing because I'm speaking too long. So, genome editing, why edit genomes? So, editing genomes is the introduction of non-random mutations. Okay, there has been methods for random mutagenesis that date back from the 1920s already, so we understand how that works, but that's not genome editing or genome engineering. You say editing when you know what the changes are. You just want a method to implement those changes in the genome. So there are 3 purposes to editing genomes. The first, 3 possible purposes, not at the same time. The first is to make the invisible visible in order to understand how cells work. Cells are really a black box. We see nothing, we just see them divide, but we don't see what's inside. And so sometimes you want to use genome editing to make the processes on the inside more visible to the researcher. You can make control changes in a genome to understand how cells work. Okay, you have a hypothesis, you say, I want to change that into that because I predict that this is what would happen. If my hypothesis is correct, then this change should happen. So in that case, you're really making a change to test the hypothesis or just to perturb the system and say, okay, when I remove that component, what's going to happen? It's going to tell me a little bit what that component was for, because now the cell has to do its life without it. And the third is to replace sequences to modify living beings according to human needs, and that's the one that everybody thinks about. Editing genomes to, grow more food, to cure diseases, or things like that. But that's just one of the 3 reasons to edit genomes. So how did this start? The very, kickoff of genome editing was in 1970 with restriction enzymes. Something that seems to be completely boring nowadays was actually probably the most important revolution in biotechnology of all times. Okay, restriction enzymes and DNA ligases were the very first technology to engineer DNA. They are still widely used today. So you probably know what restriction enzymes are. We haven't spoken about them very much, but these are enzymes that cut the DNA on particular sequences. And so the very first that was discovered is ECO1, so Escherichia coli strain R1, and that cuts the DNA on the sequence GAATTC. So why was this such a revolution? It's because before restriction enzymes were available, there was really no way to touch DNA, to do anything with it. You could purify it, and that's it, and run it on a gel, nothing else. But once you have the scissors and once you have the glue with the ligase, then you can start making recombinant DNA, and you can start creating new sequences from sequences that already exist. So that now seems completely trivial, but bear in mind that in 1970 that changed literally everything. That was the beginning of a new field that you could call biotechnology or genetic engineering. So we still use the very first techniques that were in use at the beginning when the field was born, and the one that is the most frequently used is molecular cloning. So don't get confused with cloning of an animal or a plant. When you say molecular cloning, and when I say cloning in the lab, I'm always talking about a plasmid. So plasmids are the standard containers for engineered DNA. They are small, chemically stable, and easy to amplify in E. coli. The presence of antibiotics resistance gene is exploited to make sure that bacteria contain the plasmid of interest and amplify it while they grow. You get a lot more than in a PCR reaction when you grow a plasmid. So one of the first plasmids that was discovered is PBR322. And remember plasmids, there are these like round circular pieces of DNA that are quite short compared to the chromosome of the bacteria, and they usually contain antibiotic resistance genes. So bacteria can exchange plasmids, which makes them resistant to antibiotics. That's the usual vector of antibiotics resistance. And this one contains two antibiotics resistance genes, AMP-R, so resistance to ampicillin, and TET-R, resistance to tetracycline. Okay? So the way it was used in the very beginning is that people realized that there's a BAMH1 restriction site, and it's unique in the plasmid in the middle of the tetracycline resistance gene. So they would cut with BAMH1 their favorite gene of interest. Of course, you don't know exactly what you cut when you just digest with BAMH1 the whole genome, but you can then purify the fragment of interest. And using ligase, you can ligate it together with the plasmid. Okay, so you're kind of inserting here in the BAMH1 fragment your favorite gene of interest if you have used the BAMH1 restriction enzyme to cut it as well. Like that makes compatible ends that you can ligate together. And that's really the beginning of molecular cloning.
Okay, so I have a question. So BAMH1, that's the restriction enzyme, right?
Yeah, BAMH1 is the restriction enzyme, exactly.
And, we use that enzyme to make part of the CAT resistance section and put gene in there?
Yeah, we cut here, we open the plasmid like that. There's only one cut, so you know that opens like this, not like this. And then with these two ends that are now free, you can ligate any other piece of DNA that has BAMH1 ends. And so with the ligase, then you recircularize the whole thing. That makes a bigger plasmid, and you have broken the tetracycline resistance gene.
And does that mean that this bacteria will no longer survive in tetracycline anymore?
Does that mean that the bacteria will no longer survive on tetracycline? Exactly, and that's the point. Okay, because it will grow on ampicillin, because it has the resistance against ampicillin, but not on tetracycline. So you transform the bacteria with the plasmid, you grow them on ampicillin to make sure that they have the plasmid, and then you pick the colonies and you put them on tetracycline. And if they don't grow, they probably have your favorite gene. If they grow, then the gene is intact, and so somehow the circle was closed without trapping your piece of interest. Okay, so that's a cheap way of testing the high possibility that the plasmid is the one you want. Otherwise you have to do genome sequencing, DNA sequencing, which takes more effort. If you just have to take a toothpick, pick your colony, put it on tetracycline and see if it grows, that costs literally nothing, and that takes a day. Okay, so that's faster than anything. So that was the first idea of how people started to do molecular cloning. Why use ampicillin at all? It's because bacteria, when they grow, can lose the plasmid. Okay, so you have to maintain selection, like the plasmids can just get lost. So you maintain selection with ampicillin so that when that happens, they die. And only the ones that have the plasmids can survive, and the ones that have the plasmid will amplify the plasmids as they grow. So that creates more plasmid. So once you've got your sequence of interest on a plasmid, then you can do whatever you want. You can have fun. You're God already. So using PCR, for example, with smart primers, it is possible to engineer mutations on the inserted gene, deletions, substitutions, or insertions. So these are the way that we do it in general. You have a plasmid here that contains your gene of interest, and the figure is a little bit too small, but you can see that there's a primer here going from the right to the left, another primer going from the left to the right. So because it's a circle, you're going to amplify the PCR product like this, and you're going to leave out that piece here. If you remember how PCR works, the part that's in orange is just not going to be amplified. So you have now a linear fragment at the end of the PCR, and if you use ligase, you're going to circularize it again, recreating the plasmid in circular form, but you have removed the part in orange here because you never amplified it by PCR. You can also take primers that are not perfectly complementary to the sequence of interest. they will still anneal, almost, okay? But there is like a little bit of a place, one nucleotide where that does not anneal perfectly. But that's going to be good enough for the PCR to work. So you do the reaction the same way. You amplify the circular product to get a linear one, and the part that's in blue here is the one that you designed. It's no longer the one that was on the plasmid. So your PCR product here has not exactly the same sequence as the sequence of the plasmid. And then you recircularize it again, and you get the mutation of interest here at the place where you want. Okay, that's all based on the idea that we've seen in the beginning that only the 3' end of the primer needs to be perfectly annealed. The 5' end, you have some flexibility. Okay, so if you put the mutation close to the 5' end, your primer is going to anneal where it matters on the 3', and the 5' can be anything. And you can also insert sequences using the same rationale. So if you put some new sequences on the 5' end, they don't need to anneal at all. Like they'll do the sketch that I was just doing now. You amplify the product. You have a new sequence that was present in the primer but not in the plasmid. And when you recircularize everything, then you have added that sequence that was in the primer. Okay, that goes a little fast, but think about it carefully, and you'll see that just by smart primer design, you can locally change your plasmid here, the sequence of your plasmid, according to the needs that you have. Anyway, that's not what people do. Nowadays, researchers use gene synthesis services. You pay 50 cents per nucleotide, and you receive the sequence of your choice within 2 weeks or so. So that's what everybody does now. But what people do in these companies sometimes is that, because it's just that they have people doing this and who are good at it, so they can do it for money. But nowadays, nobody really mutagenizes stuff themselves. They just pay because it's so cheap. Once you've got your modified piece of DNA, you need to put it back inside the cell. Okay, either you need to put it in the bacteria so that they can grow the plasmid, or you need to put this inside a cell so that it can express the gene of interest. And that has been a very long and difficult road in molecular biology to find ways to do that. And there are methods that work pretty well now, and they're not really understood. And when I say that, they're not understood at all, actually. So one of them is electroporation. Here you just put a very high voltage for a short period of time, and somehow that makes holes in the plasma membrane of the cell. That's an electron micrograph here of the plasma membrane, and that's before electroporation. It's totally flat, and during electroporation it creates those holes where the plasmid or the DNA can come in, just making holes for a couple of milliseconds, something like that. Another method is calcium phosphate here, and so people have got not really any idea how this works. This was the first method that was discovered. People say that creates kind of aggregates with the DNA inside and like these little razor blades that are going to just cut their way through the membrane. But it's not really clear what happens. It's just an idea that the calcium phosphate will be like little crystals. They'll be pointy and they'll just break inside the cell. And because the DNA is inside, then it's going to bring it. It works quite well if you optimize it, but people never really understood how that works. I think this is good enough anyway. As long as it works, we're happy with it. So that's transfection. These methods are usually called transfection. There are more, but I'm not showing everything. So transfection is the generic term for the introduction of DNA inside a living cell. Okay, it can be called transformation as well. But when you say transfection, it kind of means I'm taking a plasmid or some piece of DNA and putting it inside a living cell. You can also inject, but you need the cell to be relatively big, and that's the case, for example, for an oocyte here. So you can actually take a pipette and then directly put inside the pronucleus the plasmids of interest. You hold this with the pipette here that will hold the cell in place, and then with another one you inject the injector here, the DNA directly inside the cell. So once you can do this, you can start having fun. And the first thing that you can do is Gfp Fusion. So Gfp stands for green fluorescent protein. It is a protein from jellyfishes. It fluoresces in green when illuminated by blue light. It is often used for gene fusions in order to track gene product in the cell. So you have your gene of interest here, which is represented in gray, and using the techniques that I've described before, you make one protein that has one part which is the GFP here, and is in fusion with the protein of interest. So the protein of interest is going to do whatever it was doing before. This part is relatively small, it's usually separate with a tail. It doesn't really bother the protein most of the time. Somehow it was empirically discovered that having this little tail here does not bother very much. So the protein is going to do its whatever it's doing, and especially go wherever it usually goes. Okay? Then you take a blue light, so you shine a blue light, and the GFP here is going to emit fluorescence. Fluorescence is the emission of light at a different frequency than the light you used to illuminate the protein. So you illuminate in blue, and the protein becomes green. So it's a green fluorescent protein. And so when you do that, for example, you can see that very beautiful patterns appear. If this was a component of the cytoskeleton in the cells here, for example, the microtubules, then you're going to have the GFP all along the path of the microtubules. That's going to make these very beautiful looking shapes that are here. So you can see where the microtubules are in the cell. Basically, it just tells you where that protein is in the cell. That's very useful to be tracking things in space. And things don't have to be protein exactly. You can do a little bit more than that. So the infection stages of malaria were fully discovered in 2007 when researchers tagged the parasites with GFP and followed their fate in mosquitoes and in mammals. So you know malaria, this parasite that goes from mosquito to humans. So infects the blood and then goes to the liver, and from the liver goes back in the blood very regularly. And so when the mosquito feeds on the blood of the person or the mammal that has been infected by malaria, then the mosquito gets infected with the parasite as well. The parasites move to the salivary gland, and then the mosquito can inject the malaria parasite again to another person. And so until 2007, people had kind of, reasoned, understood where the parasite goes at different stages, but it was not completely sure, not completely clear. So what they did is to use this GFP. They just tagged with GFP one of the proteins of the parasite, the protein that's expressed all the time, so that when they see something that shines like a diamond in green, that has to be the parasite. And with that, they could really follow where the parasite goes, in which cells of the liver, in which cells of the blood, at which time exactly. So these were mostly understood. There was not really any news there. It was mostly in the mosquito side that things were quite different from what people had anticipated before 2007. Okay, so the point is that you have a tracker with GFP. You can just see stuff. That's what I meant with the first option in the purposes of genetic engineering, is that you can make the invisible visible. And once you see it, it becomes much easier to study. Another thing you can do with that is to add more than one color. And one example I'm going to give is rainbow. So it's a very beautiful term with a rainbow because you've got so many colors on brain. So the rainbow method randomly expresses exactly one of several colored fluorescent proteins in neurons. It could be green fluorescent protein, yellow fluorescent protein, red fluorescent protein, etc. But the neuron will express exactly one. This way it is possible to track them and visualize their connections. You can see here that when you do that, then it becomes possible to identify individual neurons, because when two neurons cross, the colors don't change, so you can really continue tracking them even when their axons cross. And so that way you can make connectomics, like to make maps of the brain in just one single experiment, because each neuron has its own color. So these are very simple tricks like that make things that used to be impossible 10 years ago now much easier to do. So this is one concept that can be pushed a little further using GFP. And you don't always want to follow things in space. Sometimes you want to study the promoter. And so in the reported gene approach, a promoter of interest is cloned upstream of a visible marker like GFP or luciferase. The construct is inserted in the organism. The cells emit light only if the promoter is active, showing where and when the gene of interest is expressed. Okay, so now you're no longer interested in the protein. You remove it completely, actually. But you want to know where and when is expressed that protein. That's what I want to know. So all we need to do is to say, I'm taking the promoter of the gene of my protein of interest, and I put it upstream of GFP. So there will be GFP production when my gene of interest is expressed. Okay, they have the same promoter. So the rationale is that if my gene of interest is expressed, then the promoter is expressed, then promoter is active, then the GFP or the luciferase that I've put downstream of it will be activated, and I will be able to see light when and where my gene of interest is expressed. The first time it was done was in tobacco, and people have used here a promoter that was specific of the circulatory system of tobacco here, and they have put luciferase. So they could see that tobacco was creating all these glowing light only in the cells of the phloem here. Okay? Not just the phloem, apparently, that's the root as well. So with this system, you don't care about the protein, you just want to know about its regulation. Where and when is my protein expressed? I don't need to do any fusion with the protein, I just throw it away. I put my visible reporter, like GFP or luciferase, downstream of its promoter. Looks pretty cool, indeed. That's the very first. That's a picture from the 90s. Now it looks a little bit outdated. People do much cooler stuff these days. You can even buy, I think, GFP fishes these days. They've been made with a zebrafish. Turn off the light, put in blue light, and then your fishes are green fluorescent. So examples of reported genes. They were used to study the development in order to know the expression patterns of identity genes. So that's the case of Drosophila here. You see they're using a reporter gene that's blue. Doesn't matter where it is as long as it's easy to see. Okay? And the gene of interest here is something that specifies the front versus the rear of the animal. So that's the embryo of Drosophila just a couple of hours after hatching, so very early. And you could see that that gene here is expressed in 2 stripes. It's a 2.5 stripes. Another gene of interest is expressed in 1 stripe in the middle. And this one here, which is particularly striking, is expressed, and this one as well, is expressed like in a zebra pattern. Okay? So you know that these genes have got these very specific patterns of expression because you've used the reporter gene to know where their promoters are active. And so wherever you see blue, that gene here is expressed. And these are genes that actually specify the segmentation of the embryo in the very early days. They're also used in virology to easily detect infected cells by inserting the reporter in a virus. So that's taken from my lab. That's what I often do in the lab, is that we have this modified HIV that has a GFP inside. So when you infect the cells with this virus, the cells become green. So that's super easy to know that they're infected. Otherwise, it'd be quite challenging to know whether they're infected or not. So whenever you see a green cell, like these little dots here, if it's green, it's infected by HIV. And you can easily sort them based on color, and then you can get cells that are all infected and leave the rest away. So we're going to make a little break now. Let's make the break first, and we'll take questions if there is any after the break. So let's take a 5-minute break, if that's okay with you. We meet again at 3:08. Let's make it 3:09. Okay, welcome back. Are there any questions with what we've seen in the first half? Do you understand the concept of reporter gene? Okay, that with the reporter gene, you're studying the promoter or the regulatory regions. Okay, you're not studying the protein itself. We carry on? All right. So let's speak about transgenics, like the way you actually make a transgene. And transgenes are genes acquired after fertilization. Okay, like there are the genes that you get from mom, the ones that you get from dad, and there are your transgenes. They are the ones you get neither from dad nor from mom. So they must be delivered and maintained in the target cells. Most of the time, but not always, they are carried and delivered on so-called vectors that are viruses or transposons. so that they integrate at a random site in the genome. So vector is a little bit of a very general term in biology. So when I was second year student and the teacher kept using the term vector, I said like, what is a vector? Just define it for me, please. And he just mumbled something, and I do the same, just mumble. Like it's not really defined what a vector is, but it's called a vector because it means that it carries a gene of interest. So that can be a plasmid, a transposon, a virus. And the idea of vector is that it will carry that gene from one organism to the other. That's what's a vector. Okay, so don't get confused with the term vector. It's a very generic term. When people don't want to specify which platform they use to make the transgenic, they will just use the term vector to say it doesn't matter. And if it matters, they will be specific. They'll call it a transposon, a plasmid, or a virus. So here's an example with a transposon, the piggyback transposon. And if you remember, we've seen that transposons are naturally occurring selfish elements. There are some in our genome. It's about 3% of our genome. They are much more prevalent in prokaryotes, in bacteria and archaea. And they make copies of themselves by jumping around. They don't really copy themselves, they more cut themselves and paste themselves. So if you put the transgene here on a plasmid, and you have these blue sequences that are specific for the piggyback transposase, the piggy base here, then if you put that plasmid together in a cell with a transposase, transposase is going to cut the blue bits there and then mobilize completely at random that particular sequence and place it somewhere in the genome. So that's one way that you can insert the gene from a plasmid to the genome of interest. Okay, you need to bring in that cell together the plasmid, the transposase here in pink, and then the magic will do the rest. So what's the problem if you just have the plasmid and you don't have the transposase? What do you think is going to happen? Why is that not a super good idea if you want to do transgenics? Like the main, yeah, go ahead if you have an idea.
Sorry, I was going to ask you to repeat the question.
The question is, if you don't put the transposase and you just put the plasmid with your gene of interest, the transgene of interest, why is that not such a good idea to not have the transposase? Yeah.
So like the plasmid will get lost like after something degraded?
Yeah, the plasmid will get lost, and will it get degraded? Probably not. It's more like the cells divide. Okay, like you have one cell that receives the plasmid, but the cells keep growing, and because there's no origin of replication in the plasmid for eukaryotes, then the cells will just not replicate the plasmid. And so you have two cells, only one of them has the plasmid, then 4 cells, only one of them has the plasmid, then 16 cells, and 32, etc. And still one cell has the plasmid, so it's not going very far. We want to put this in the genome so that when the cells divide, they also multiply the transgene of interest. Okay, that's why you do that. You can have a transient transfection, it's called, when you just put the plasmid, but you want a stable transfection. You want this thing to be inserted in the genome. So that's why usually you need a vector that has also a component that will make it go inside the genome, like a transposon or a virus as well. That's what viruses do, like HIV. So transgene vectors are plasmids that contain a virus or a transposon, often with multiple cloning sites and a resistance gene. Example, neomycin. So that's one example here, P-Lexin vector. So you have the origin of replication for bacteria. That's the reason why you can amplify this in E. coli with the techniques that I've presented before. There's an ampicillin resistance gene here, again for bacteria, because you need to select the bacteria that have the plasmid. So you put them on ampicillin and they can grow because they have the plasmid. And this here, that's the part that's for mammals. Okay, you have the LTR here, the long terminal repeat. So that's MMLV, MMSV. So that's a murine virus here for mice. So the part that's here is for mammals. The part that's on the left here is for bacteria. But because this plasmid will also be used in mammals, then all these things are for the mammalian part of life. And there's the neomycin gene here that will be used in mammals, not in bacteria. So the gene of interest is cloned. The MCS stands for multiple cloning site. means that you have multiple restriction sites. So you can choose the one you like best, HPA1, XH01, BAMH1. You usually have more than that, like around 20 or something like that. Then you cut exactly at that region the plasmid, you put your gene of interest, you close it, and now you have a virus, okay, that has like the viral sequences on the left, the LTR, and on the right as well, the gene of interest and the neomycin resistance gene. So the gene of interest is cloned viruses are produced by transfection, cells are infected, and transgenic cells are selected with neomycin. So let's review the process. You first make, with the recombinant DNA technique, this plasmid that has your gene of interest that you use with restriction enzymes, okay, that you put with restriction enzymes. With that, you can transfect cells. Once you transfect cells, they will produce the virus, because that's a virus here. Okay, so that produces an RNA that corresponds to that bit here, and that's enough to produce new viruses. Remember that when a virus is produced inside a cell, it will produce more virus. So it does not matter that you did not start with an infection. You can start with a transfection. Once you produce virus particles, well, you produce viruses. And once you have viruses, you can then infect cells with it. Okay, so that's a multi-step process. You first transfect, collect the viruses because they just go out of the cell, and then use these viruses to infect a new cell with it. And when the cell is infected with the virus, then they will actually do what viruses do, like HIV and this type of viruses, which is insert themselves in the genome. Okay, multi-step process. There is the transfection to produce viruses, and then the infection to infect the cells with a stable transgene. And you put neomycin at the end on the second cell to make sure that they have the virus, that they have been infected by the virus, and that the gene, including neomycin here, has been put on a genome, and when they divide, that they have the resistance to neomycin. Okay, so that's typically the way it's done, with a transfection, then an infection, and then a selection for the resistance. So that's the story of Randy the mouse. The SRY gene guides the formation of male organs during early development. In 1991, researchers put an SRY transgene in an XX mouse, turning it into a phenotypic male. So, of course, if you're XX as a mammal, then you have to be female. But there was this idea that the reason why you can be male is just because there is this one gene, SRY, that in reality females have got everything that's required to be male, except one gene, that there's just one gene that's required to be phenotypically male. And so to prove that, or at least to test that, what they did is that they took a female XX mouse and they just expressed this one transgene in it with the technique that I've described. It's not exactly the same, but that's the idea. Okay, so once this SRY gene was inside the female embryo with just one cell, then that embryo has developed exactly like a male with everything. And so that's the that's Randy here showing off his parts. Thank you, Randy. And so otherwise, even though that's an XX mouse, it's phenotypically exactly like a male. It's sterile, you could not have babies, but it looked pretty much like a male with the hormones, etc. So that kind of proved the point that really there's only one gene that triggers the whole cascade to become a male from the one cell stage. And that gene is of course in the Y chromosome, and that's why where you have the Y chromosome, then you're a male. Y chromosome also contains one or two extra genes that are used for the sperm to actually be mobile, and so that's why this mouse here is infertile. Even though that looks like a male, the sperm were not viable. So the researchers injected plasmids containing SRY into embryos. This was sufficient to obtain a viable male mouse, showing that the plasmid containing everything that is needed to specify the male identity. Okay, just one gene is sufficient when you're a female to turn the whole development into male. And so they worked out the cascade of signalization here is because all the other components of this message for our maleness, if you want, is already present in females. All you need is that one signal, and that's the dominant signal. If you send it only once and early in development, then the rest of the development will be all male. So another kind of transgene which is quite useful is inducible transgenes. Inducible transgenes are transgenes that are expressed only when the experimenter wants to. Induction systems must be cheap, fast, and easy to deliver. One of the most popular systems is based on induction by tetracycline, which is taken from bacteria. So tetracycline is an antibiotic, and the way bacteria resist to antibiotics is that they have a gene that just counteracts its action. But they're a little bit smarter than that, in the sense that when there is no antibiotic in the medium, bacteria don't produce that protein because it's a waste of energy. So they produce the protein only when they can sense, when they can feel that the medium contains tetracycline. So it's a two-component system with a resistance gene that will inactivate tetracycline or allow the bacterium to grow in the presence of tetracycline, and another component which is sensing the presence of tetracycline and activating the first gene when it senses tetracycline. Okay. So in practice, people don't use tetracycline, they use doxycycline here. Doxycycline is a more potent version of tetracycline. It's a little bit more stable in solution. Tetracycline dies fast, otherwise with doxycycline you can keep it for 2 days in the medium. Tetracycline is only for about a day. So you just use doxycycline, but that's the same thing. So there are two components in that system. So here it's a little bit small, sorry about this. There's your gene here, which is indicated in blue. So you have an interest in producing that transgene, but only when you want. Like you want it to be off, but at the moment when you say go, you want the protein to be expressed. So how do you do that? You clone your gene downstream of this green promoter here. And the green promoter contains the elements that are required to respond to tetracycline. Okay, that's the DNA that's required to respond to tetracycline. And you also use another gene, so there's a second transgene, which is written like the TET-on3G gene. And so that's the one that produces the sensor of tetracycline. Okay, so you need that sensor, otherwise you'll not be able to know when there is tetracycline in the medium. So the sensor is that blue protein here. So when tetracycline or doxycycline is in the medium, there is the formation of a complex between that blue protein here and tetracycline that activates the complex, that then recognizes the promoter and activates transcription. Okay? So there are two parts. I'll do them in other, I'll swap the order. The first is a sensor of tetracycline that activates transcription when there is tetracycline. And the second is a promoter that's keyed to that sensor upstream of your gene of interest. So with that system, you need to have two components, the sensor and the promoter of tetracycline upstream of your gene. So if both of them are in the cell, then you just need to put tetracycline and your gene of interest will be expressed. Is that clear, that when you put tetracycline and doxycycline in this case, that will activate your gene here? Right. So because there are two components, that's a little bit complicated, fairly annoying. If you have to put two transgenes at the same time, every time you want to do these experiments. So the way people do it is that they use genetic. So they actually have a mouse here that produces the blue part here, and they have a mouse here that has the other part. On the sketch on the left, they are showed as they are on the same chromosome, but it does not have to be the case. So this mouse they can keep all the time. That's the donor mouse. They just need to, like your gene can be anything, and for example, every PhD student in the lab would have a different gene here. And they would all cross it to the same mouse that is there. Okay? Because that's the one that gives the sensor of tetracycline. And this bit here is really the gene that you're studying. So you cross the mouse with the yellow part, the mouse with the blue part, and then you get a mouse that has both components in all of its cells. So now when you give tetracycline to that mouse, here is going to express that gene. Okay? So you don't need to make complicated transgenes every time. When you need to have two transgenes in the same cell, usually you do it with genetics because then it's much cheaper. You just need to maintain these mice, and if it's a male, you can actually get the children for 3 years with only one single mouse. Okay, is that clear? That you need these two components there? And TRE, that is here, stands for tetracycline responsive elements. That's a special sequence in the promoter. This is recognized by that protein in blue here, only one is bound by tetracycline. So that's what makes it responsive.
I was wondering, in the TET-on3G gene, is it transfected into the cell?
So is the TET-on3G gene transfected into the cell? The answer is, in that particular case, no. Like the very first time, yes, it was a transgenic, but when you created that mouse here, after you have the mouse, you don't need to, because the best way to bring genes to a cell is fecundation. That works much better than transfection. Just let nature do its business, present Mr. and Mrs. and then they do the combination by themselves. So that's the way it's done in this case. When you can't do it this way, you do, because it's always cheaper. But in order to get that mouse, yes, it was done with a transfection in the very beginning. So another tool that got a lot of attention recently, from 10 years ago onward, is optogenetics. It's a very spectacular set of techniques. People who developed it got Nobel Prize. Many of the things actually that I presented today got awarded Nobel Prize, starting with the restriction enzymes, for instance. So channel rhodopsin is an ion channel that opens when stimulated with light. It is used to engineer select neurons so that the experimenters can activate them at will and see the effect on behavior. So that's a transgene that looks like that. That's a pore on the membrane, but that's a very, very special pore in the sense that when it receives blue light, the pore opens. And in this case, in the case of CHR2, it lets in Na plus and Ca2 plus and lets out K plus. So that brings depolarization of the neurons of the membrane. So that will actually create an action potential that will activate the neuron. So the neuron will do its normal work, whatever it's doing in the normal life of the animal. But when you shine blue light on it, then it becomes active. So you can kind of overrule whatever state was given by the other neurons, because when you shine a blue light, you activate it. So you see sustained light here, you see the action potential that are firing like crazy. You can also make pulses, like every couple milliseconds, and then you get an action potential just after the pulse here. You can also do the reverse with the NPHR channel, which lets in Cl minus ions, so that will hyperpolarize the neurons, which will inhibit them. So you have this particular neuron here, which fires all the time, but when you activate NPHR, you hyperpolarize it, and then it becomes like flat, no activity whatsoever. And as soon as you release the light, then the neuron goes back to normal life. Okay, so you see what this is doing is that here this is a mouse that has on the head implanted a little light device with a cable here. That's probably an optic fiber that allows the experimenter to activate the neurons of interest whenever they want. So they can see, if I activate the neuron in that particular context, what's going to happen to the mouse. So that tells me what that neuron is doing for the behavior. Okay, so that's a very useful tool, even though that looks a little bit cruel from that picture. But that's a very useful tool for the neuroscientist to be able to dissect the role of the neurons in particular behaviors. You cannot study everything with that because the poor mouse is kind of attached with a wire, so it cannot go everywhere. So studying movement, for example, is a little bit of a problem, but you can still study a lot with this thing. You can also study this without so much hassle in flies, for instance. You don't have to have a cable, you just to put a blue light on top of them. And if you're really a little bit smart, you can express that blue protein only in some kind of neurons, not in all the neurons of the brain, of course. And for that, you would need to use a promoter that expresses the gene only in some key neuron that you'd like to study. So with the combination of both the activating and the inhibiting, people have been able to understand much better what are the roles of some of the key neurons in the brains of mice. I don't think this has been done in humans at all, and I don't think it's planned to be done in humans at all. That's a very impressive technique. So another one which is really important to know is the technique of knockout. Now we've seen how to make transgenes, so you want to add genes to the genome, but of course the other thing that we can do, which is equally useful if not more, is to remove a gene from the genome. So how is that done? Traditional gene knockouts were based on recombination, and that started around 1990. A BAC clone, which is the same as a plasmid but much bigger. A plasmid is like 10 to 20 kb, a BAC is more like 100 kb. So a mammalian gene usually doesn't fit on a plasmid, you need a BAC. A BAC clone is transfected in embryonic stem cells, ES cells, that are selected with neomycin and ganciclovir. So here again, the sketch is a little bit small, sorry about this, but how does it work? To make a knockout, you don't need to remove the whole gene, you just need to remove one exon. Okay, that's sufficient, because if it's the key exon that contains the catalytic side of the protein, then it's not going to work, or it's going to make a frame shift and the protein is going to be dead. So the idea is we're going to just remove exon 2 here of that particular protein. And the way they do this is that they have this construct in a BAC, so think plasmid, if BAC is not to your taste. And we have the sequence of exon 1 here, the sequence of exon 2, and we have replaced, with the techniques that I showed in the first half, exon 2, we replaced it by neomycin, so the resistance gene for neomycin. And after exon 3, we put a gene for thymidine kinase. So when we do the transfection, what we hope will happen is that there will be a recombination, ectopic recombination between exon 1 here and exon 3 together. So when that happens, they will actually put the DNA of neomycin in place of exon 2. Okay, that's what we hope will happen. That doesn't happen very often, but that's what we hope will happen. And so that's the desired outcome, that exon 2 is gone here and neomycin is present. So these cells, when that happens, are resistant to neomycin. We can just select these cells on neomycin and they will tell us that they have received that. The problem though is that this double recombination is relatively rare, and that particular construct can also integrate somewhere else in the genome in bulk, or even sometimes in tandem, like you would have 5 or 6 insertions in a row of the BAC. And if that happens, because they got the whole package, they got the thymidine kinase gene at the same time as the neomycin gene. And when they have thymidine kinase, then they become sensitive to ganciclovir. So you can kill them with ganciclovir. So if the cells are resistant to neomycin and to ganciclovir, it means that they have neomycin resistant gene and they don't have thymidine kinase. So they're much more likely to look like that. That's the trick they use. It's a fairly complicated idea, especially compared to what we do now, but that got the job done here. So one of the 2 alleles is knocked out. This double recombination is already super rare, so you can't hope it's going to happen twice in the same cell. The cells are injected in a very young embryo. Okay, and so that's the way they created the mouse that is here. Okay, it's by first transfecting an ES cell, embryonic stem cell. Then the embryonic stem cells are selected on neomycin and ganciclovir. Then they are injected in the embryo. So that's not the same as the injection of DNA here. These are cells that you see here, and that's an embryo, a very young embryo. So it contains, I don't know, let's say 30 to 60 cells in the embryo, and you put another, obviously, 4 or 5 that are coming from this ES cell culture. Okay, so these ES cells have the knockout, and you're putting them in the embryo. That produces a camera with 2 different colors. So that produces mice that look like that, because some of their cells are the ES cells that were injected with a pipette here, and some of the cells of the mouse are the ones that were here. And of course you take cells from mice with different colors, and you can easily tell that they're a chimera, because then they look like that. Okay? With a bit of luck, ES cells colonize the gonads, and the children of the chimera have the heterozygous mutation. So it doesn't really matter what the skin color is or the fur. What matters is the gonads. Are they like these cells? Because if they are, it means that the children of this mouse will have the knockout. If the gonads are as they were before, like as the donor mouse, then the children are just wild type. They're not interesting at all. So you're looking for this camera here. You can find them easily because like they have two colors. It means the experiment worked, but you still need to make sure that their children have the knockout. And if they do, bingo, you're done. Okay, with a back cross, you can obtain a double knockout. So the children of this mouse, with a bit of luck, are heterozygous knockout. And then you take the children of this mouse, cross them again with daddy, and then you can get double knockout. Like that. Okay? So it's a long process, basically. That's approximately 3 years of work for several talented people. So that was something that at the time when I was doing my PhD, when someone said, I'm doing a knockout, you say, okay, good luck, see you in 3 or 4 years. That's the end of your PhD. You won't even have the time to analyze the phenotype. You will just have the time to do the knockout. Okay, the process is relatively straightforward but long, and each of these steps is prone to errors as well. So the first success was obtained around 1990, and that was a major success for genetic engineering. So of course nowadays we don't do it this way. As you imagine, is we're using CRISPR-Cas9. There's all the fuss about this technique. So what is it exactly?
Your mic.
The mic is not working? 1 2 1 2. No, it's dead. So I think the battery is gone. Yeah. Yeah. Okay. Now my voice is too loud with all that electric energy. So, CRISPR-Cas9 is an immune system against phages that was discovered in archaea and bacteria. Actually, it was discovered in archaea in the beginning. Cas9 makes a double-strand break DNA in specific sequences that are complementary to a 20-nucleotide RNA. So that's the sketch that you've got here. There's the gRNA for guide RNA, and the protein Cas9, which is represented in the background here, is driven to the site of interest by the guide RNA. That's why it's called the guide. And Cas9 has the job to cut the DNA on both places, here and there. That's what Cas9 does. The system was repurposed for gene knockouts and knock-ins as well. An exon of interest is targeted, cut, and repaired in a sloppy way that creates a double knockout in a single round. If we compare to the previous slide, here we target exon 2. If we can remove exon 2, we consider that the gene is gone. So we do the same thing here. But we design a guide RNA here that just has the sequence of exon 2. And if we can bring this Cas9 complex inside the cell, then they will actually cut exon 2 here on both strands. But this is so efficient, they will cut allele number 1 and also allele number 2 at the same time. Contrary to this old method that use double recombination, which is super rare, this is super frequent and super efficient. Okay, so you get 2 cuts, and then when it's prepared by the pathway that is called non-homologous end joining, this is a pathway that tends to be very sloppy. So it adds and removes nucleotides so that you're going to have a frame shift most of the time, plus a couple of deletions of key amino acids. So the exon is going to be totally messed up here. It's messed up one way in allele number 1, messed up another way in allele number 2, but it doesn't matter. Both ways are messed up. So the exon is gone, and you can say that the protein is gone. So that's the base strategy for CRISPR-Cas9 gene knockout. Okay, so basically you do this here, that step, but you knock out both exons in a single step without even needing to select for neomycin and gangcyclovir. So it's more like you just fast forward that step with one single experiment. And you make it more efficient because you got the double knockout already at the very beginning. Okay? So the system was repurposed for gene knockouts and knock-ins. An exon of interest is targeted, cut, and repaired in a sloppy way, that I read. Previous methods were available, but the cloning steps were more difficult. So the only part that you're really increasing a lot with CRISPR-Cas9 is the very beginning of the process of gene knockout. You still have to get the children, etc., and you're still making these chimeras. Okay, that hasn't changed. So one still needs to inject mouse embryos and select chimeras, but the process is much shorter now. Like you need only 3 months to get to the chimera, probably even less. And so that's much shorter than having to wait 2, sometimes 3 years with the previous methods. So another way you can use the system is you don't want to like just destroy the exon. You want to do a knock-in. It means that you want to add some sequences. You want to replace them with something that is a little bit more engineered, in the sense that you want to really give a particular sequence there. So what you can do is you get this double cut here that is just created by Cas9, and you can provide also together in the cell donor DNA that contains sequence on the left which is identical to this one, sequence on the right which is identical to the other one, and in the middle you put some new sequences, the ones that are in blue. And what you hope is that there will be a double recombination just like before, and that will actually replace the sequences here where the cut was by the sequence in blue. So have we really gained something? I said that here this double recombination is super rare. So if we need this double recombination here again, is that really a gain? And the answer is yes, because double recombination is rare if you don't have a cut. But when you have a cut already, when DNA is already broken, the cell is actually quite likely to choose to do this double recombination, because that's one way you can repair the ends of the DNA. Okay, so that's much more likely to happen because you have made that cut, and that's why the CRISPR-Cas9 system was used in the first place for. So one issue is how specific is CRISPR. You're going to cut all over the place to make double strand breaks of the DNA, so you're going to create multiple knockouts in reality. If the guide RNA is not 100% specific, you're going to have cuts in the genome. So yeah, okay, you're going to get the genetic engineering of interest, sure, but what about you're going to get 20 other mutations in the genome that you didn't want? So that was always one of the big issues with CRISPR. And nowadays people kind of accept that this is the case. This is not that much of a huge deal, because the method is just so much cheaper that people say like, we're just taking these extra mutations, it's worth it. But still the question of the specificity is an important one. So the Pam sequence is 5' and GG3' for Cas9. It is not present in the guide RNA, but it must be present in the target RNA. So if I show it here, like there's the Pam sequence here, so you see it is present in the DNA that must be cut, but the part in green in the RNA doesn't have it. Okay, that part, the Pam sequence, is in the target but not in the guide. It is the anchor point for Cas9 to open DNA and for the guide RNA to anneal. The original CRISPR-Cas9 technology creates many off-target cuts. That was a bit of an issue, even though it's fairly specific. Bear in mind that you have this triplet here, NGG, and N is not specific at all. And you have then 18 to 20 nucleotides, so that's not extremely specific. Like you can have mispairing of these 20 nucleotides to several places in the genome. And so the system has been a little bit improved in a sense. The modified Cas9 has only a nickase activity. It doesn't make a double strand break, it makes a single strand break. That's called a nick. Like one of the strands remains intact, but the other one is nicked, like that. And can create more accurate cuts when two guides are used together. So people often use this modified Cas9 now that won't really cut double strand break, that just make a nick. And if you use two guide RNAs that are bringing the two Cas9 very close to each other, that's the Cas9N here for nickase, you can see that you've got this guide RNA that will bring here Cas9 on the left, the other one that's going to bring Cas9 there, And if you got one nick here and one nick there, then the amount of DNA that's kind of holding the chromosome together is not enough. It's going to break like that. But if there is a stray Cas9n that goes somewhere and just makes a nick, it's not going to be a big deal. The nick is going to be sealed, and not much going to happen. You're going to get a double strand break only when the two guide RNAs are together with the two Cas9n. Okay? So why the Pam sequence cannot be on the guide RNA? So the guide RNA is produced from DNA. Okay, there has to be something that's transcribed. And so if you remember that the system is coming from bacteria, like there at this CRISPR, it's really the locus where these guide RNAs are produced, clustered regularly interspersed palindromic repeats. That's what it means. And so if the Pam sequence was present in that locus, then it would cut itself all the time. Okay, so that's as simple as that, is that you need to find a way so that the system can cut the incoming viruses, but without cutting the genome itself. If the Pam sequence were in the locus where the RNA are produced, then Cas9 would keep cutting the CRISPR locus all the time, and that would destroy it. So that's why this sequence is assumed to be present in the virus, but not in the genome. So the CRISPR-Cas9 technology can be used to create gene drives, or gene drive system, where the transgene spreads through a population in a non-Mendelian way. So how does that work? What's the idea? Is that you create a transgenic animal that has Cas9 here, the guide RNA, and also the cargo. It means whatever you like. Like it can be GFP, there can be a protein to cure something, whatever that is, doesn't matter. The cargo is really your wish as a genetic engineer. And the guide RNA is made in such a way that it targets the homologous copy of that allele. Okay, that's the modified allele after it's been modified by the genetic engineer to have Cas9 and the cargo. And the Cas9 targets the unmodified allele. So if both alleles are together in the same cell, what's going to happen? The guide RNA is going to be expressed, Cas9 is going to be expressed, and so that is going to bring Cas9 to cut the other allele. Okay? The other allele is going to have a double strand break, and like I said, that's going to trigger, quite likely, a double recombination. So it's going to use the sequence here and there to repair the broken DNA, and it means that it will actually copy the transgene from one allele to the other allele. So that's a transgene that copies itself from allele to allele. That's what's called a gene drive. And so here, if you have normal inheritance, that's you have the allele, one allele from this particular fly, let's say, and then according to Mendelian law, it will be present only in half of the progeny or something like that, okay? Because it's one of the two alleles, not all the children can have it. It's only half of them. But if you have a gene drive system like this one, what's going to happen is that you're going to first convert the other allele inside that fly here. So it goes from heterozygous to homozygous with the cargo. Okay, and then it means that once it's homozygous, it will pass the allele all the time. Okay, because when you're homozygous, you always pass the same allele. So the children of this fly are going to be heterozygous because they get one copy from, let's say it's the dad, and now they get a copy from the wild type flies, but they will go from heterozygous to homozygous again. Okay, so as soon as a fly is created, as soon as it's heterozygous, it will convert to homozygous with that system. So if you have one copy of the transgene, you get two copies of the transgene. And when you have two copies of the transgene, all your children have the transgene. So it means that in just a couple of generations, all the flies in the population have the transgene. Okay, whereas if you have just a normal system like that, the fly, just one lineage would have the transgene, and that would not be a lot. You know, there's no chance that it would take over the population. So So such systems were produced. They were proposed to be used to engineer natural populations, like mosquitoes in particular, to make them extinct or resistant to malaria. But they were never used because there is no undo button. Okay, you understand that when you do that, it's gone. Like when you start to have this hyperproliferation of that transgene, you can't press Ctrl Z. So people thought, of course, it looks like a good idea, but you never know. And so for now, ethical committees say, just hold your horses. We're not doing anything. So another technique based on CRISPR, which became quite popular recently, is CRISPR scarring. CRISPR scarring technologies consist in producing multiple cuts in an array of identical transgenes. Example, GFP arrays. You put plenty of GFP, like 20 of them. Each cell repairs the DNA in a different way. So you have one guide RNA that will target 20 copies of GFP, and each GFP is repaired in a different way with this sloppy system, so that the daughter cells can be traced as belonging to the same clone. So early on in the development, here in the cell lineage, you create, let's say, you have 20 cells only at that point, and you induce the CRISPR-Cas9 system. So each of these 20 cells will just break the GFP array in a separate way and repair it in a separate way. But then, after the repair is finished, it's fixed. Like it stays like that forever, which means all the daughters of these individual 20 cells will keep exactly the same scar from Cas9. Because once the DNA is repaired, it's no longer modified. And just by looking at the scar they have, it can tell you which is their ancestor, because they all share exactly the same scar. And of course, that brings us to what people imagine when we say genetic engineering, gene therapy. So SCID-X1, or severe combined immunodeficiency X1, is a genetic disease where children are born without T cells or natural killer cells, NK cells. Only one gene called gamma C is defective. Okay, so that's a disease where children are born with a mutation. There's only one gene that they miss, but they have no immune system. So that's the problem. In 1999, the first gene therapy trial was conducted. And so they need to live in these bubbles. I don't know if you've seen these pictures of the babies who live in the bubble, because they have zero immune system. So the air must be absolutely clean, because even a very mild infection would kill them. They have no possibility of defending themselves. It's absolutely a horrible, horrendous life for these kids. And that's why in 1999, the first gene therapy trial was conducted, where bone marrow cells were collected, infected with a retrovirus containing gamma C, and injected back in the bone marrow of the patients. Right? You get the idea. They missed the gamma C gene. Let's give it to them. As simple as that. The patients were cured. Give it to them. Take the bone marrow cell, put it in the bone marrow cell, and put the bone marrow cells back, so that some of their cells will have the gamma C gene, and that's enough for them to have an immune system. The patients were cured, but they got cancer, all of them, because the retrovirus inserted into unactivated oncogenes. So there was not enough thinking about what would happen in that case. They used a very old generation retrovirus, which is not used nowadays anymore. And that older generation had the problem that the viruses activate cell proliferation genes. So the cells that were supposed to be just bringing the gamma C gene did that, but on top of it gave cancer. One patient died, but the other 15 were saved and cured of SCID-X1. Okay, so of course they hadn't really thought this through, and so that was a bit of a setback. But the ones that they could save, once they could get rid of the problem of the cancer, at least that worked, because that's indeed all that was missing for these kids. It was the gamma C gene, and if you give it to them, then they have a normal immune system and they can get out of the bubble. So it was, let's call it a mixed success. Some people were very happy because you saved 15. Some people said, no, you kill one, and that's really a stop. And people were quite divided at the time about whether we should go through with gene therapy or not. And people I know were quite angry that the person who did this used very, very old technology that they already knew were outdated at the moment when they did this. So another way that this has been used, which is quite similar, is HIV therapy. The only patients cured of HIV received a bone marrow graft from healthy donors with double null CCR5 mutation. Okay, some people in the population have got this double knockout of the CCR5 gene. CCR5 is a co-repressor of the surface of the cell, co-receptor, sorry. Deficiency of CCR5 makes patients immune to HIV, at least to this kind of HIV. If you don't have CCR5, this type of HIV cannot infect you. They don't even have what it takes to enter the cell. So only a few cells are resistant to HIV, but this is enough for the immune system to develop and suppress the virus. Okay, you get the idea? So it's not really a gene therapy. Like these people had HIV, and so they say, you need a bone marrow graft. Let's at least give you a bone marrow graft from someone who's immune to HIV, so that when you survive the graft, you will produce immune cells that are resistant to HIV, and so you will have an immune system. It's not a gene therapy. It's just that if we're about to do the graft, let's at least give you a graft that's going to cure HIV, in the sense that it will give you an immune system. So this has worked 2 times, if I remember, this method. But this inspires a possible gene therapy where CCR5 is removed from bone marrow cells that are re-injected in the patient. This has not been done, but you see the idea. It's the only way that people so far has been cured from HIV. There's, I think, 1 case of spontaneous remission, which is not fully understood. But the plan is, let's take your cells, we knock out CCR5 from it, and then we give back your cells. But the problem is that bone marrow transplant is an extremely dangerous procedure with only, like something like there is a 20% chance that things go bad in bone marrow graft. So they won't do that to everybody, and that's also very costly. So the idea is in the air, it's not been done, and it cannot be scaled up that much. And the treatment for HIV is actually working, so people prefer to keep taking pills rather than risking a 20% chance of dying. And then that brings us to the CRISPR baby scandal, of course. So in 2019, I'm not sure how to pronounce his name, this must be He announced the birth of twin babies where he used CRISPR-Cas9 to mutate CCR5 so that they would be immune to HIV infection. So they didn't have HIV. There was no reason to do that, but he thought, I'm going to give them the gift of life. And so this person came to the fertilization clinic, and I think without even consulting the parent, he thought, let me make a gift to your 2 babies. I'm going to remove the CCR5 gene from them using CRISPR-Cas9 so that for any reason these 2 babies will not be able to be infected by HIV. So in his description of the fact, he said there was a very good act, but in 2020 he was sentenced to 3 years in prison by the Chinese government. It's not completely clear whether he's actually serving this time, but there was such a condemnation by the scientific community worldwide that this was done without an ethical committee that he had to take some prison time for having done that. So that's what I wanted to say about genetic engineering. So the key points to remember is that most genome edits are now technologically feasible. They're even cheap-ish, thanks to CRISPR-Cas9. The CRISPR-Cas9 technology made edits cheap. So it's really important to understand that there's nothing that we could not do before CRISPR. Really. CRISPR just made it cheap and fast. And so, except maybe the gene drive idea that really could not be done without CRISPR very much. So it created new ideas, etc. But mostly it's because things that used to take 3 years are now 3 to 4 months. And so that's really all this is. People say, oh my God, this is a huge revolution. Yeah, sure, in terms of money, that's huge. But in terms of concepts, it's not really a huge deal. Edits of the human genome are still limited, of course, because there are ethical considerations. Are there any questions or things you'd like me to explain again? Yeah?
For the gene therapy for the children without the T cells, did they update it, or did they just leave it when they found out that they got cancer? And then
So the question is, what did they do after they found out that the kids have cancer? I'm not quite sure. Of course, there was no kind of cancel button on this transgene here, but now this is what they do. They have if you want the same idea as the tetracycline gene, something that you can just kill the cells that have the transgene with one compound. Like you have a cancel button that you put inside the transgene. That's now in routine. People say we want to be, just to make sure that we can remove these cells by giving a product to the patient or something like that. Back then there was nothing. If the kids are cured of SCID, then they must have had the gamma C gene. So the transgene must still be there. I think they just removed the cells that were cancerous by just treating the cancer, and the ones that were not cancerous were allowed to stay, if I remember. It's a good question. Other questions in the chat. I have two questions regarding the gene knockout. Does double recombination use CRE-LOX-P, and why is RE not used in making KO restriction enzyme? No, it doesn't use CRE-LOX. This is natural recombination that happens just like in DNA repair. And why not use restriction enzymes? Because you cannot. This is inside a cell. You can't express a restriction enzyme inside a cell. You can express it only outside in the tube. If you put this in a cell, you're going to chop the genome into small pieces. So can't use restriction enzymes in vivo, only in vitro. Another question, how rare are we talking about for recombination? What are the chances? So it's kind of hard to give you a number because people don't really count the cells that they have in the beginning and the cells that they have in the end. But if we go back to this slide that you're both referring to, this one here, so typically this is something that you take millions of cells, and if you get 5, you'd be considering yourself very lucky that this thing happens 5 times, so that you have 5 shots at creating this chimera mouse here. So a handful, like that's something like 2-3 years of work, and you'd get 5, something like this. This is how rare we're talking about.
For the gene drives, can you clarify why it's duplicated the cargo?
So in the gene drive, why is the cargo duplicated? Same reason as here, okay? Like you have this cut that triggers a DNA recombination. And so if you have DNA recombination, if you cut here, the only way you can recombine is by using the other allele as a template. And because it's going to use the blue bits here for the recombination, that's going to add the bits in the middle when the DNA is repaired. That's the mechanism. All right, we're 2 minutes past time. I think people are waiting to get in the room, so we need to leave. Thank you very much, and see you next week.